Microphone?
You might hear me, yeah? So in my talk, I'm going to try to cover, or I've asked to present two things. And the first thing were to give some background and sort of understanding for you what the challenges are, both for AstraZeneca and for this sector and the industry in general. And then connect that to how could research done at the ESRF or the MAX or the ESS type facilities play or make a difference for facing that challenge. And it might not be obvious for you, but I hope when I take you through this trip that there are some clear, very obvious connections, how we need to work together, not least to create these wonderful facilities. So AstraZeneca, we are a big company, and talking of billions of dollars, we invest about $5 billion each year in research. So we are a big player, but we do so in very many fields. And that is one of the challenges. The drug discovery is a very complex and long process, and we need to have a lot of both internal and external knowledge feeding into the process to actually make it work. So we're active in all three major areas of pharmaceutical development, and that is we develop both small molecule drugs, biologicals, and also vaccines. And we do so in three disease areas, and they cover some of the most important disease areas where you still have a lot of unmet medical need. And those are cancer, cardiovascular, gastrointestinal, infection, neuroscience, and respiratory and inflammation areas. So what are the challenges? And if you consider lucky to be in the industry and sector that have a lot of challenges, we certainly are lucky. But not all of them are that pleasant. Talking for the industry in general, we have got to receive most of the revenue, most of the sales comes from what is called blockbuster drugs. These are drugs that sell a lot of use by a huge group of patients, and therefore generate a lot of revenue. But a lot of these ones are running out of patents. And I will come back to some of the challenges behind that. The other area which has been on the broadcast is safety. There has been some spectacular withdrawal of medicines when they've been shown there have been side effects like Vioxx. And if a company enters into that area and have a safety problem, it almost can be the end of that company. So we need to be really careful and to make sure that we understand what our drugs are doing and that are safe with patients. And also most recently, and this is sort of the heart of my story, is that there's also a cost factor coming in here. As the patient or the population grows older and we suffer a lot more disease, the overall cost for the healthcare for elderly and the whole population is going to basically explode. So there's going to be a lot of discussions around cost-benefit things, around the use of drugs and the use of treatments, which one can we afford and which can we not afford to have. And it's very important that we can prove that we can create the medical or the value to pay back the money that we get from selling our drugs. If you look at this slide, it can look like everything is just going fantastically well. We have a huge increase in sales. We have large spending in R&D, but if you look at the lower bar, it's actually decreasing in the number of new medicines coming out onto the market. So despite increased spending in R&D, we are creating fewer drugs than ever. And there are several explanations for this. One is that the easy stuff has been done. For those diseases for which there are simple mechanisms and easy targets to work with, we generally have drugs now. What are remaining are much more complex diseases for which we might not even know the disease mechanisms yet. And it's going to cost us enormous amount of money and research actually to be able to develop medicines in those areas. Some financial magazines actually advised us to move out of R&D completely. With your success rate and the enormous cost, it's not a sustainable model. You should leave that to others and just focus on developing or selling generic drugs. Our CEO, David Brennan, just a few days ago actually made a statement saying that we are going to stay in the business of being in sort of innovation-driven, R&D-driven, and we're going to make our money from prescription drugs that are still under patent. But in order to do so, we have to start to do a lot of things very different. And we have to focus on R&D in many ways. We need, beyond all, start to build strong collaborations with the rest of the healthcare systems. We need to understand what the payer are prepared to pay for. What the to really know that the drugs we're developing are going to sell or bring back the revenue. And who are these guys we need to collaborate with? And there's a long list of people that we by necessity need to collaborate, and that includes also governments, it includes academics, and it includes other businesses, even our competitors. So some recent examples we've had where we came up, or we found a drug, or a competitor of ours found a drug that's useful for a disease for which they didn't have the machinery or the knowledge how to develop it and get it onto the market. In that case, we seek collaborations, even between companies who are normally competitors, just to get it as cheaply and as quickly onto the market as possible. We buy companies to get knowledge into the company, and we in-license things in the different stages of research, trying to tap into what's already done out there so we don't invent the wheel several times, because in the end, it's actually the taxpayers and the healthcare system that pays for these drugs. We're also looking increasingly, and that's why the bar is biggest, to the academic community for collaborations, not least to get access to the research facilities we're here to discuss today. Discovery is a long process. It takes about, in the best of times, 12 years to get onto the market. And what you normally consider as research is only the blue bit here, is the initial start, the discovery bit. But then we have to spend a lot of effort just proving the value of those compounds we're developing early on, that they are safe, that they're effective. And just to give you an idea, I think in the blue sector we have hundreds of projects, and very few of them actually moves into the next phase because we're successful in finding a drug that can, we expect to have an effect on that disease. And as the process goes on, only very few actually makes it all the way through onto the market. So the cost, because there are a lot of products in the early one and there are a few in the later stage, is about equal. And the total cost to get a drug on the market is well above $1 billion today. So for the cost of building an ESS, you would probably get about one drug to the market. So predicting things early, to have a more predictive science, so we know we don't waste money in bringing things into extensive studies that will not show out to have an effect. And they might even get to the market before we understand that they have some side effect and therefore have to be drawn. It will save huge money. So we invest a lot in predictive science, and a lot of the areas of predictive science is actually research that could also be done at these facilities, including imaging, develop disease models where we can follow the progress of the disease and understand the mechanism. But this drug discovery is not an easy process. There are lots of factors that you need to take into account, the parameters to refine as you go through. And I could spend the next 2 or 3 hours just explaining this sketch, but I will not. I will just simplify it and said, to go from early in vitro studies into studies in humans, it's all about molecular interactions. And molecular interaction, we already heard through the slides here, is a key. It's the heart of drug discovery. And you need to understand, and we're using a plethora of technologies in the company to try to understand how our drugs interact with their target molecules, kinetically, energetically, and even geometrically, to develop their characteristics. Right, so the dominant factors that are the property we're using is used in protein crystallography and NMR to get the binding modes. It's ITC and technologies to get the thermodynamic profile, and some SPR technologies to get kinetics. But if you really want to look at binding, to make it simple, you can see there's some binding which we find good, and there's some binding we find not so good. So in the left-hand side, you have enthalpy, and enthalpy usually comes from matching van der Waals interaction, hydrogen bonds very neatly together. You get a binding through enthalpy. Entropy is a more unspecific type of binding, which usually comes from dissolution effects, and that is hiding hydrophobic surfaces that don't like to be in water. So they prefer to jump onto the drug target and sit there. The disadvantage with entropy bit is that it's usually very unspecific. If you really want to be specific, and you do want to be specific in drug discoveries because you want to avoid side effect, that things are interacting with a lot of other targets in the cell. So enthalpy is good. Understanding enthalpy, though, and where it comes from and how to engineer hydrogen bonds is not always that easy. So the technology we tend to use is the one on the left-hand side, which is we travel to ESRF for MaxLab and we collect data, and you get an electron density distribution, and you can build a model of how the drug interacts. But if you look at the water molecules, they're round. And water molecules should really look like Mickey Mouse because there are two hydrogens sitting on them, but they're invisible to X-rays. But if you go to neutrons, it's a different picture because they interact with the nuclear and you can neatly see the protons. And if you do a clever experiment like they've done that you exchange with deuterium, so the most acidic one will be a change, you can actually see that the one on the sitting between the two nitrogen atoms up there, I can't point because the laser is not working, has been exchanged. It's actually deuterium ion, so we can know it's acidic. So all kind of neat experiments can be done to increase understanding of fine. So to tune off, how important is it to get right when you do your drug design? So these are a family of statins. These are drugs taken by a lot of people, probably in this audience, to lower your cholesterol levels. And they are in the order. You can see the first on the market. One of the first ones was Lescol, and then the other ones followed. And we were last on the market, and you can wonder, it's good to be last on the market. If you look at the blue bar, you can see our drug is the most potent one. And that is good. But furthermore, if you look at the green bar, you can see that it gets most of its binding energy from enthalpic effects. And that was our talk. Those are the good interactions with the correct hydrogen bonds. And they should be more specific. And hopefully also be more potent. So interestingly enough, the first one, which doesn't have any enthalpy, Lecol, not only interacts with a target to lower your cholesterol, it also interacts with hepatitis C viruses. So it's a very non-specific binder. Biocol, the second one, is actually withdrawn from the market or had to be withdrawn by Merck because of huge side effects. Lipitor has been the blockbuster drug that probably most of you still eat today. While Crestor then was the last one on the market, but it has shown some quite spectacular results. So this is just a couple of weeks ago when the FDA approved this drug to be used for a group of patients which is have an increased risk of cardiovascular disease, CVD. And if you look at the last line, you can see that by eating this or taking this treatment, and this is the only statin that had shown this effect, you halve the risk of dying in heart attack, stroke, or arterial revascularization. So I hope I convinced you that it might be worthwhile to really do your homework and get to a drug that has the right profile, and you need to know what you're doing, the mechanism you're going for, because then you can reach results, which means all the difference. I think if you look back in the previous slide, there was a question mark on the headlines of Lipitor, where there was actually a cost-benefit having that kind of treatment in patients. I think if you can show these kind of results, that you halve the chance for people of dying, I think we can really say that there is a cost-benefit. And to get there, in many, many cases, you need to use information that becomes available through biophysical studies, including structural studies, for example, at ESS, or hopefully at the ESS when it gets built, where we can see neutrons more readily, protons more readily, or hydrogens, but also the MaxLab facility that will come online shortly. It's very much at the heart of this drug discovery at this point. So we'll end there on this journey, which took us through the challenges we have and some of the ways we're trying to mitigate those risks and challenges by sort of bringing new technology into the drug discovery process.